For decades now, we have been hunting for this biological check engine light of aging.
Oh, yeah. The absolute holy grail of the field.
Right. We've been looking for that one undeniable marker that proves a cell has basically gone zombie, what we in the field call a senescent cell.
Exactly.
You see it in all the longevity literature, right? You find the marker, you target the cell, you cure the disease, but there is a new landmark paper that just dropped, and it proves that this single universal marker, it actually doesn't exist.
No, it really doesn't. And that is a massive paradigm shift for us.
It's huge. So instead, scientists actually had to rely on machine learning to find this hidden pattern.
Yeah, because the diagnostic waters of aging are just incredibly muddy. And this paper, which is published in Molecular Cell, it just wades right into the deep end of that mud.
Which is exactly what we are going to unpack in this deep dive. Now, for anyone who is already following the aging field, you probably know the basics of the SASP, right?
Right, the senescence-associated secretory phenotype.
Yeah.
It's a mouthful.
It really is. But basically, we know these zombie cells stop dividing, but they don't actually die. They just sit there.
Yeah, they sit there and they secrete this highly toxic cocktail. It's full of inflammatory cytokines and growth factors.
Which just wreaks havoc on the surrounding tissue. But what fascinated me right out of the gate with this paper, the SenCat project, is the sheer scale of how they tried to track this SAC down.
Oh, the scope is unprecedented. SenCat stands for Senescence Catalog, by the way.
Right. And they didn't just look at, a couple of lung cells in a petri dish. They essentially built a microscopic human.
It really did. Because to definitively prove whether a universal marker exists or not, you have to test everything.
So how many cell types are we talking about here?
They analyzed 14 primary human cell types.
14.
Yeah. We are talking about everything from WI38 lung fibroblasts and BJ skin fibroblasts.
Which are pretty standard.
Right, standard stuff.
Yeah.
But they also looked at astrocytes in the brain, melanocytes, endothelial cells from coronary arteries.
Wow, so the heart.
Exactly. Plus bone marrow mesenchymal stem cells, skeletal myoblasts, and even preadipocytes from fat tissue.
Okay, so they essentially have a cross-section of the entire human body. But they didn't just let these cells age naturally on a shelf, right? They forced them into this zombie state.
They did. They systematically pushed these diverse cell types into senescence using 4 distinct mechanisms or paradigms.
And the why here is super crucial for you to understand because the trigger matters.
It matters entirely. So the 4 triggers were CTIS, which is chemotherapy-induced senescence.
Okay, chemo.
Right. Then IRIS, which is ionizing radiation-induced. OSIS, which is oxidative stress-induced. And finally, OIS, which is oncogene-induced senescence.
So just to break that down, the reason you use all 4 is because the initial insult completely changes how this cell reacts.
Precisely. Think about the physical mechanics of the damage. Ionizing radiation, like X-rays, literally shatters the DNA double helix. It's physical trauma.
Right.
But oxidative stress, that's more like rust. It degrades the cell's lipid membranes and the proteins from the inside out.
So radiation is a sledgehammer and oxidative stress is rust. What about the other 2?
Well, chemotherapy chemically blocks cell division. It jams the gears. And an oncogene is it's a hyperactive cancer gene. It pushes the cell to replicate so unbelievably fast that the cell panics and hits the emergency brake.
Oh, wow. So because the initial damage is completely different in each scenario, the cells cry for help. Its biological footprint looks entirely different.
Exactly. So they map all the RNA, all the proteins, across 14 cell types and 4 damage triggers. They're looking for the holy grail marker.
But they don't find it.
They didn't. The massive reveal of this huge data set is that there is no single universal marker for senescence.
Which is just crazy to me.
I know. Not one single RNA transcript or protein was consistently altered across every single cell type and trigger.
Okay, wait, I have to push back on this a little bit, because what about the gold standard markers?
You mean the ones everyone uses?
Yeah. If you read any paper on aging, they always point to P16 or P21 or the loss of the nuclear envelope protein LMNB1. Are you telling me those pillars of aging research just failed?
Well, I wouldn't say they failed entirely, but their reliability it's completely context dependent.
How so?
For instance, canonical markers like P21, they were upregulated in many models at the RNA level.
Yeah.
But when you actually looked at the physical proteins they encode, the end product, they didn't even crack the top 20 shared markers.
Wait, really? The protein wasn't even in the top 20?
Nope. And LMNB1, it did consistently drop, but the degree to which it dropped, varied wildly.
Like depending on the tissue.
Yeah, depending on if you were looking at, say, a skin cell damaged by radiation versus a lung cell damaged by chemo, it's highly variable.
Okay, so if I'm trying to visualize this, it's like we've been looking for a universal zombie uniform so we can spot the undead in a crowd. We assume they all wore red hats.
Right, just look for the red hat.
Exactly. But it turns out zombie skin cells wear completely different clothes than zombie lung cells. And a cell zombified by radiation is dressed entirely differently than one zombified by oxidative stress.
That is a perfect analogy. You can't just scan the crowd for a red hat because half the zombies are wearing blue scarves and the other half are wearing yellow jackets. The uniform is totally dictated by the tissue type and the trauma it endured.
So if we look at what we've established so far, testing 14 human cell types against 4 distinct damage triggers proves our traditional biological check engine lights are basically fundamentally flawed.
They are.
Which leaves you, the listener, with a really interesting question. If you were a scientist and your trusty single-gene markers just failed you across the board, where exactly do you look next?
Well, the SenCat researchers realized they had to look deeper into the multi-omic reality of the cell.
Multi-omic, meaning multiple levels of data.
Exactly. Specifically, they compare the transcriptome, which is all the RNA. Think of it as the biological instruction manuals.
Okay, the blueprints.
Right. They compared that with the proteome, which represents the actual physical proteins built from those instructions.
And here is where the biology gets a little mind-bending for me. Because if RNA is literally just the instruction manual to build proteins, shouldn't the RNA markers and the protein markers match up perfectly?
You'd think so, right?
Yeah. If I have an instruction manual for a chair, I should end up with a chair.
In a perfect linear system, yes.
Yes.
But biology is rarely linear. What's fascinating here is the incredibly complex, multi-leveled regulation of gene expression.
Meaning things get messed up between the blueprint and the final product?
Exactly. An RNA transcript might be created, the manual is printed, but it could be rapidly degraded by the cell before it's ever even read by a ribosome.
Oh, wow.
Or the protein is successfully manufactured, but then it's immediately modified. It's folded differently or maybe destroyed by the cell's internal recycling system.
So the cell changes its mind.
All the time. Because of this massive post-translational regulation, the SenCat team found that the overlap between the transcriptomic markers and the proteomic markers was shockingly low.
How low?
Less than 20%.
Less than 20%. That means 80% of the time, the instruction manuals and the final physical products in these senescent cells don't even align. That is incredibly noisy data.
It's totally chaotic.
Mm-hmm.
And because the individual genes and proteins were so noisy, the researchers realized they had to stop looking at individual parts.
They had to zoom out.
Right. They started looking at broader cellular pathways. Think of a pathway as a cellular assembly line.
Okay, an assembly line.
Even if the specific workers on the line, meaning the individual genes, even if they change from cell to cell, the assembly line itself is still running and producing a result.
And this is where the data suddenly gets super clear, doesn't it?
It really does. Even without a universal uniform, these zombie cells absolutely share conserved behaviors.
Like what kind of behaviors?
Well, the RNA data showed massive, consistent upregulation in the p53 pathway.
Which is like the master regulator for DNA damage, right?
Exactly. And also NF-kappa B, which is the major driver pumping out that toxic inflammatory SSP cocktail we talked about earlier.
Right, the poison they spit out.
They also saw cells dramatically ramping up their lysosomes.
Which makes sense because lysosomes are basically the cellular trash compactors. These cells are heavily damaged, so they are trying to break down their own broken proteins just to survive.
Exactly. But the proteomic data revealed something even more profound. They saw widespread metabolic reprogramming and the heavy activation of something called the EMT pathway.
Ah, EMT, epithelial to mesenchymal transition. This pathway is wild.
It really is.
If you picture standard epithelial cells, they are like rigid bricks in a wall. They are stationary, they are tightly packed, and they hold the structural integrity of your organs together.
Right. They have these tight junctions locking them in place. But when EMT activates, those rigid bricks fundamentally change their physical properties.
They lose their grip, right?
Yeah, they lose their tight junctions. They become mobile, fluid, and squishy. They essentially revert to a primitive state that is designed to migrate toward a wound and remodel the damaged tissue.
So if I'm tracking this right, let's pause for a second. We are at the 5-minute mark of this deep dive, and to really lock this in, we need a way to remember this shift.
Good idea.
If you want to remember what actually defines a zombie cell, think of the MED list of zombie behaviors. MED. D.
I like that. So M is for metabolic reprogramming.
Right. Changing how it generates and consumes energy.
E is for EMT.
Losing its rigid shape to become mobile for tissue remodeling.
And D is for damage response.
Activating P53 and pumping out inflammation. So MED. Metabolic EMT damage.
That perfectly encapsulates the shift in understanding here. Senescence is defined by shared biological behaviors, not shared individual markers.
So we've moved from looking for a single smoking gun to looking at the entire crime scene, the metabolic shifts, the tissue remodeling. So knowing this, think about how your own body responds to a physical cut or like a deep injury. Knowing about this fluid tissue remodeling EMT pathway, does it make sense that these zombie cells might actually just be cellular paramedics whose programming got stuck on a permanent loop?
That is the exact biological tension we're looking at. They're trying to heal the tissue, but they never actually finish the job.
They just get stuck.
Yeah.
Yeah.
But recognizing that behavioral tension, it doesn't solve our core diagnostic problem. We still need a reliable mathematical way to actually find them in a living body.
And here's where it gets really interesting. Because human brains and traditional statistics couldn't find a universal marker in that 80% noise, the SenCat team brought in the heavy machinery.
Machine learning.
Yes, they brought in AI.
They utilized a highly sophisticated algorithm known as a penalized logistic regression model.
Penalized logistic regression, that sounds intense. What does the penalized part mean?
The term penalized is key here. It means the math actually punishes the algorithm if it relies too heavily on any single gene.
Oh, wow. So it can't just cheat and look for P16.
Exactly. It forces the AI to look at the entire orchestra of genes rather than just listening to one really loud instrument.
That is brilliant. And the way they trained it was also super clever, right?
Yeah.
They used a leave one cell type out strategy.
Precisely. It's a cross-validation strategy. So they would train the AI on 13 of the cell types and then test it on the 14th cell type that it had never seen before.
And they just rotated this over and over.
Over and over. This ensured the AI didn't become biased toward any single cell lineage, like a skin cell or a brain cell.
Right, because otherwise it would just learn what a damaged brain cell looks like, not what senescence looks like universally.
Exactly. It forced the algorithm to find the deep, hidden mathematical weights of various RNAs and proteins that consistently predicted senescence across everything.
And the AI actually found them. It flagged specific, robust markers that traditional biology totally missed.
It did. For positive predictors, meaning things that go way up in zombie cells, the AI heavily weighted a protein called VDAC2.
Now, why would it flag VDAC2? Because VDAC2 controls the pores at the mitochondria, right?
Yes, it literally regulates how the cell manages its energy and its stress.
Which ties perfectly back to that metabolic reprogramming we just talked about in the MED mnemonic.
Exactly. The AI saw the behavioral shift mathematically. It also flagged inflammatory RNAs like CXCL8 and another one called GDF15.
And it didn't just look for things going up. It found powerful negative predictors, too, things that consistently shut down.
Right. For instance, TOP2A mRNA absolutely plummeted.
What does TOP2A do?
TOP2A is an enzyme that helps untangle DNA during replication. So imagine DNA as a twisted phone cord. When a cell divides, it has to untangle that cord.
Okay.
But if a senescent cells permanently lock in cell cycle arrest and it's never ever dividing again, it has no need to untangle its DNA. So it just shuts that gene off.
That makes total sense.
It also downregulates things like SERPINH1, STMN1, and various histones.
Histones being the spools that new DNA wraps around.
Exactly. No division, no need for new spools.
So instead of a simple pass-fail test looking for one gene like P16, the machine learning created a weighted score. I like to think of it like a FICO credit score for your cells.
Oh, that's a great way to look at it.
Right, because your credit score isn't determined by a single late payment on a water bill. It's a complex algorithm weighing your credit utilization, your payment history, the age of your accounts.
Right. It takes dozens of factors.
Exactly. It takes dozens of small financial behaviors to give you one highly accurate, undeniable number. And the SenCat score takes dozens of these gene expressions, weights them perfectly, and gives you a definitive zombie score.
And this score proved incredibly accurate. They validated it on external data sets. Entire groups of cells the AI had never been trained on, like IMR90 lung fibroblasts.
And what happened?
The ML score perfectly mathematically separated the senescent cells from the healthy ones.
That is wild.
Yeah.
So by completely avoiding the bias of single markers and using this leave one out AI algorithm, they created this highly accurate, weighted FICO score for cellular senescence.
They really did.
Which brings up a slightly uncomfortable question for you listening. How comfortable are you with the idea that an AI algorithm might actually be better at diagnosing the deeply hidden aging mechanisms in your own organs than a human biologist?
It's a paradigm shift we
Oh, yeah.
basically have to embrace. But to prove this AI score actually matters for the future of medicine, they couldn't just leave it in the computer. They had to take it out of the petri dish.
Right. They had to prove it works in reality.
Exactly. They had to validate it in vivo, in living, breathing mammals.
Yes. And this is really the climax of the paper for me. So case study number 1. They took mice and injected them with doxorubicin.
At a dose of 10 milligrams per kilogram, to be exact.
Right. And doxorubicin is a heavy-duty chemotherapy drug. It's known to induce rapid systemic senescence.
So to measure the impact of this drug, they used a really cutting-edge technique called snRNA-seq, single-nucleus RNA sequencing.
Okay, single nucleus.
Right. They extracted the lungs and kidneys of these mice at exact intervals. Day 0, day 1, day 2, day 4, and day 6.
That timeline is super crucial, right? Because it allows us to watch the kinetics of senescence. It's not an instant switch where a cell just turns zombie overnight. It's a cascade.
Exactly. It's a gradual process.
Mm-hmm.
But snRNA-seq is notoriously difficult to work with. It has what we call a very high dropout rate.
Meaning the data is spotty.
Very spotty. When you are trying to sequence the RNA of a single microscopic nucleus, you often only catch a tiny fraction of the RNA that's actually there.
Oh, I see.
It's like trying to listen to a symphony on a radio station with terrible static.
Yeah.
You only hear fragments. And because of that, traditional markers like P16 completely vanish in that static.
But when they applied the weighted SenCat machine learning score to those single nuclei, the signal cut right through the noise.
It did. Because the score looks at dozens of weighted markers. If 1 or 2 drop out due to sequencing noise, the overall score still holds up perfectly.
So they could literally watch the FICO scores of the cells in the lung and kidney tissue rise day by day following the chemo injection.
Day by day. But if we connect this to the bigger picture, they didn't just stop at the rapid damage of chemotherapy.
Right, because most of us aren't experiencing sudden chemo damage, we're just getting older.
Exactly. They wanted to see if the algorithm could track the slow, physiological creep of natural aging. So they analyzed multi-tissue data from mice at 3 months old.
Which represents youth.
Right. Then 12 months old, representing middle age, and 23 months old, representing old age.
And the FICO score worked perfectly again. The ML scores mirrored the natural accumulation of senescent cells over the lifespan of the mice across multiple organs.
It was incredibly consistent.
But then the researchers went one step further into the 23-month-old data, and this absolutely blew my mind. They used a computational tool called CellPhoneDB.
CellPhoneDB is fascinating. It performs what's called ligand receptor inference analysis.
Which means what, exactly?
Essentially, it mathematically maps the biochemical conversations happening between different cells in a tissue.
So it listens in on their text messages.
Pretty much. It looks for sender cells that are pumping out specific signal molecules, which we call ligands, and looks for receiver cells that have the specific receptors to catch those exact signals.
Okay, so they looked at the aging lungs of these mice, and the tool revealed that the senescent fibroblasts, our zombie cells, were acting as aggressive sender cells.
Very aggressive. They were heavily using the TGF-beta pathway. That's transforming growth factor beta.
And they were using it to transmit signals directly to non-senescent epithelial cells, the healthy neighbors.
Exactly. And TGF-beta isn't just a friendly ping, just saying hello. It physically commands the receiving cell to alter its extracellular matrix.
It pushes it to become fibrotic and stiff.
Yes. It completely highlights the true danger of the SASP. These zombie cells are actively communicating with their healthy neighbors and effectively corrupting them.
They are shouting commands that degrade the structural integrity and the function of the tissue around them. It's literally the mechanism of organ failure.
It is. It's a domino effect.
Okay, so if we take a step back and look at what we've just covered, we're at our second scheduled recap here. A few massive realizations lock into place. Let's hit the bullet points.
Let's do it.
First, we've proven that AI-weighted multi-omic scores work in living tissue, cutting through the intense static of single nucleus sequencing.
Right.
Second, The score successfully tracked rapid cellular aging caused by chemotherapy in real time.
Yep, the day 0 to day 6 study.
Third, it tracked the slow, decades-long accumulation of senescence in natural aging.
Or across multiple organs, yeah.
And fourth, using interaction mapping with cellphone-DB, we now have definitive proof that these zombie cells actively corrupt their healthy neighbors using TGF-beta signaling.
They are actively destroying the neighborhood.
So knowing that these cells actively talk to healthy cells, physically commanding them to stiffen and fail, does that change how you view the aging process in your own organs? Because it's not just passive wear and tear on your joints or your lungs, it's an active toxic neighborhood effect.
It radically changes it. And honestly, it completely rewrites how we must approach longevity therapeutics moving forward.
So what does this all mean for us? Bringing it all together, the SenCat project is just a monumental resource. It proves once and for all that we must abandon the myth of the single magic marker.
We have to.
We can't just look for P16 and call it a day. The future of finding, diagnosing, and eventually treating aging cells relies entirely on these multi-omic, AI-weighted scores that look at the entire behavioral assembly line of the cell.
Exactly. And that brings us to a final, slightly provocative thought to leave on.
Oh, I like provocative thoughts. Let's hear it.
We established earlier with the MED mnemonic that these senescent cells shut down their cell cycle, but they drastically ramp up tissue remodeling pathways like EMT.
Right. They lose their rigid shape, they become fluid, and they migrate to fix a wound.
Exactly. They revert to a fluid state to fix that wound, but then they get stuck. They can't finish the job. So if these zombie cells are fundamentally just cellular paramedics that are perpetually frozen in the repair phase, what is the right medical intervention for the future?
Oh, wow.
Do we design senolytics to assassinate them entirely, potentially removing cells that are actively trying to maintain tissue structure?
So wiping out the paramedics.
Right. Or do we use these new AI signatures to find a way to simply unpause them so they can finish the healing job they started and then naturally clear themselves out?
That is a wild thought to leave on. Do we kill the stuck paramedic, or do we just figure out how to fix their radius?
That's the multi-billion-dollar question in longevity right now.
It really is. Well, thank you so much for coming along on this deep dive with us. Keep questioning the hidden mechanisms in your own biology, because as we saw today, the diagnostic waters might be incredibly muddy. But with enough multi-omic data and the right algorithm, we can still find exactly what's lurking in the deep.